Well, thank you very much for having me here. It's a real pleasure. As you've gotten from my introduction, well, did you mention that none of my degrees are in chemistry? Right. So my training is all as a geologist, and progressively during that career, it went from geology to geochemistry, and there's a long history of geochemistry, and in particular organic geochemistry at the University of Bristol, which is why I ended up in a chemistry department. And this makes me, I suppose that's probably the gateway by which I won the interdisciplinary award, which is sort of the reason I'm here. But more importantly, I think it's, I don't know, it puts me in a unique position because I love chemistry. I'm someone who loves chemistry not because I chose to do it early on. Actually, organic chemistry in particular caused me no end of difficulty early on, but rather because I love geology, I loved environmental sciences, but I realized to truly understand and answer some of the questions I wanted to, I needed to use chemistry. And chemistry, I still think, is probably the most powerful scientific discipline for really understanding the way the world works, because if you want to understand the way the world works, there's a lot of physics, there's a lot of geology, but there's also a lot of biology in life. And the only discipline that can really help us understand how the environment and how physics and how life all work together is chemistry. So these days, 100 years from now, 200 years from now, yes, the physicists will have their day in the sun when you can understand all of that from a truly fundamental realm. But really, I think you can only take it down to chemistry right now. And I think chemistry is a fantastic discipline, which is why it's very exciting to be in a school of chemistry. And why it's very exciting to be here, especially when I do realize I am a bit of an odd duck. So it's particularly nice to come in and talk to chemists because of that passion. Now, as you mentioned, I'm now director of this Cabot Institute. And you might think that the logical way to do this talk would be to start with what I do and then sort of culminate in the Cabot Institute, because of course it is a big multidisciplinary institute, and that is the essence of this sort of interdisciplinary career progression I've gone on. But I actually want to start with that. I want to start with that because that actually informs why I do the research I do and why I've taken on the challenges I have. So I can get that out of the way at the beginning, dispense with that, and then get into the meaty bit of the research that I do. And effectively, what are the challenges we deal with? Well, the challenges are all arising from the fact that there's 7 billion people who live on the planet, going to be 9 billion by the middle of this century. Not just a lot of people, but a growing middle class across the globe, urbanizing population, an aging population, and that imposes all sorts of impacts and demands, not just on how we live, but on our environment. We certainly need to extract more wealth from the environment. On the way over, I was talking to a geotechnical engineer on the plane, and she worked for a mining company. We need to mine the earth for our resources. We need to extract the energy that we need from the earth or from the environment. Our food is dependent upon the environment. And of course, food is a bit of a tricky one because related to food, you need water. And of course, water, you need enough of it. But as we know in the southwest of England, you can't have too much of it. So all of these are challenging issues. We need to provide the energy, the food, the water for 7 going on 9 billion people and all of the other resources that we need, all of which has to come from our planet in one way or another. And we need to do that without degrading the environment at the same time. And of course, we can degrade the environment in all sorts of ways. As many of you would know, there's vast amounts of plastics being in the ocean, plastic nanoparticles. We don't even know what the impact of those is on life yet. All sorts of different ways that we impact the environment. But probably the one that we think about the most, and the one that I've spent most of my career studying, is how we've changed the chemical composition of the atmosphere and how that is affecting our global climate. So those are all the types of issues that we want to grapple with, and that is why we set up this big Cabot Institute, which is sort of multidisciplinary writ large. And I always make the case to say, well, you know, at the end of the day, oops, where's my laser? You really can't think about our food security without thinking about water. You can't understand what's going to happen with water if you don't understand climate change. You need to think about all of those problems. And of course, you need to think about them in an interdisciplinary context with a lot of social scientists and engineers along for the ride. So that's what motivates me. That's writ large. But if I want to dig down a little bit to what really motivates me as a scientist, what I'm really concerned about, it is this climate and environmental change that's right at the heart of this. And I always tell people, I don't think climate change is a problem in and of itself. What climate change does is it exacerbates and complicates everything else that we're trying to deal with. So what I want to do is I want to try to understand either what is happening so we can avoid it, or if it does come, what will its impact be so we can be better prepared for it. So just to start out with a little bit in terms of what we actually know about climate change before we get to what we don't know and the areas that I want to work and interrogate, we know that carbon dioxide concentrations are increasing. They've been increasing for since the Industrial Revolution at a pretty remarkable rate. This little inset shows a couple of years ago when we passed 400 parts per million a few years ago. That is scientifically meaningless but somewhat symbolic threshold. And I'll come back to 400 parts per million later in the talk. We know that this is because of human activity. We know it from an economic perspective, but we also know it from a chemical perspective. We see it in the radiocarbon composition of the carbon dioxide, but we also see it in the stable carbon isotopes. You sort of see here is that we're going from values, and I'll come back to this a lot. I'm going to talk a lot about stable isotopes. We're going from a value that looks a little bit more like carbonate, inorganic carbon, and looks a lot more like organic matter. So you're seeing that shift in the isotopic composition of the atmosphere because we're burning organic matter. So we know that we're causing this increase in carbon dioxide, and we also know that it's fairly unprecedented. If we merge that historical record that we've measured as atmospheric and analytical chemists, and we compare that to, again, an equally challenging record to develop, which is that based upon the bubbles of gas in ice cores, we extend that back as far as the ice cores go, almost 1 million years. What we see is just how dramatic the carbon dioxide concentration is compared to what existed in the previous 1 million years of Earth history. So we know that we're doing something quite unprecedented. And we know it will cause warming. That's just atmospheric chemistry at its heart. We know that if you add more carbon dioxide, which is terribly effective at absorbing long-wave radiation, not so good at absorbing short-wave radiation, we will warm the planet. This here is a probability distribution of a really important term, the climate sensitivity. Climate sensitivity is how much warmer the planet gets globally if you double carbon dioxide. And we think about it in this sense because it is an exponential relationship. So this distribution comes from all sorts of climate models, all different varieties made by different countries, different groups, run under different parameterizations, different schemes. And you end up with a distribution that suggests that climate sensitivity is right around about 2.5 or 3 degrees. But there is this long tail, and there is this probabilistic range. And people spend a lot of time arguing about it because it has some pretty profound implications, which I'll show you on the next slide. But I think it's useful, before I move on to that, to actually show where that actually comes from. And effectively, what we're looking at is the impact of carbon dioxide directly, as well as the fast feedbacks in the Earth system. So we know, and this is fairly straightforward, that even Tyndall had largely sorted out well over a century ago, that if you double atmospheric carbon dioxide, you get about a little over 1 degree of warming. The first thing that happens is that means you will then hold greater moisture in the atmosphere. And since water vapor is also a greenhouse gas, that has an immediate and direct positive impact on warming. But then it gets complicated. You might melt snow and ice, and that will add to more warming. Or you might have clouds if you have more water vapor, and that will then have a negative effect. It will reflect more sunlight, and you will have cooling. And you put this all together in the climate models, and the emergent property is something like this climate sensitivity that you get out. Different models get out different numbers. That's why you have this range. And what does the range mean? Well, if we only look at this, which is basically business as usual in terms of carbon dioxide emissions up until the end of the century, what it means is that the warming of the planet, the whole planet, parts of the planet will warm a lot more than this, others less, will be anywhere from about 3.5 to 5.5 degrees. That is a big range for the entire planet. That is a great deal of uncertainty. And this propagates all the way through the climate system and the biological systems that will be affected by this. Will we warm the planet by 3.5, 5.5 degrees? That affects how much sea level will rise. Will it rise by about 0.5 meter or 1 meter? This, again, is a probability projection that is largely dependent upon how much you warm the planet. Of course, it has a lot of other uncertainty. Of course, you've got the uncertainty in warmth, but you've got all the uncertainty associated with how the ice responds and the thermal expansion of the ocean. And this even does not really consider a mechanistic understanding of how the ice sheets will melt. So there's a lot of uncertainty in this projection that is not even shown in this figure. If we move on and begin to look at what really matters to people, what really matters to people is it wet or dry? That's drought or famine. That's flooding. How much it rains really is going to be the most important factor in what controls whether or not people live or die. Now we're really stuck in sort of the realm of atmospheric physics, but I'll come back to the environment, the chemistry in a few moments. But I like this because it really highlights the limits to our knowledge. What you see in blue are areas where climate models predict it will get wetter, and areas in these hot colors are where it'll get drier. All models predict the same things. Dry gets drier, wet gets wetter. However, if you look in detail, you see these parts of the map that are stippled here. This is where 2/3 of the climate models agree on just the sign of change. All the areas that are not, we don't even have consensus amongst the models of whether or not it gets drier or whether it gets wetter. And if you're concerned about drought and famine in sub-Saharan Africa, that is a problem. If you're concerned about global food production, much of which is coming from California or the central part of the United States, that is a problem. And I don't really know where the really cutting issues in terms of climate change are, but my gut is it's not so much how hot and dry it is. It's not even how wet it is. It's going to be food. Can we provide food for 9 billion people when we don't know this at a fundamental level, what's going to happen to the rainfall? So we've got this deep uncertainty. Now, this slide that's going to come up will make us feel rather smug as chemists because the chemistry is relatively straightforward. In terms of if you add carbon dioxide to the atmosphere, it will dissolve in the ocean in fairly well understood patterns, and that will decrease the pH of the ocean. This is how much it's decreased so far, not too much, and here's how much it will decrease again if we continue having CO2 emitted to the atmosphere as we have. So that's good, right? The chemistry is fairly straightforward. The thing is, I don't actually care about chemistry per se. I care about the biogeochemistry. So the question is not what is the pH of the ocean in the future. The question is how does that affect life in the ocean? What is the biological consequence of lower pH? And that is deep uncertainty because we need to think about how a lower pH, more CO2, higher temperature, and all of these other factors will combine to affect an organism. But of course, we don't care about an organism. We care about an ecosystem. So how will all of these things come together? And this is a realm of deep uncertainty. All of this is basically preface for showing you the range of uncertainty that we have when we're trying to understand what will happen in the future. Our tools are imperfect. And you basically have here sort of a scale moving all the way from things that we do understand but have largely probabilistic forecasts like temperature or sea level change. Those factors for which we have insufficient understanding, so we largely know what will happen to ice sheets, but there's lots of questions. So we can put a number on it, but large error bars. And then moving on to where we have deep complexity or systemic uncertainty, especially in biological systems, where there is no forecast. There's no forecast for what will happen to many of these biological systems that actually integrate all of these different factors. So this is the world in which we live. So how do we understand those? What are we doing? Well, what we do right now is we observe the Earth system and we're trying to understand the consequences. How has temperature changed over the past 100 years? How have the ecosystems changed? How has sea level changed? How has the ice sheets changed? How are all of these things happening? But this is not satisfying as way as science goes. It's necessary, but it's not satisfying. We want to know the answers to this experiment before we conduct the damn experiment, right? I mean, sometimes people say, well, you know, as a scientist, what do you think we should do? I said, well, as a scientist, burn all the fossil fuel. Burn it all and let's see what happens, right? But that's not necessarily, you know, the most humanitarian option, right? So this is what we do. We try to, it's like turning on the hob and then measuring the stew temperature after a couple of minutes and trying to then forecast what might happen in the future. And basically, you know that the stew will get hotter, but aside from that, you have some pretty fundamental questions. The other thing we do is we try to create artificial planets, right? We try to create them in models. We try to make climate models is no longer an appropriate term. These are Earth system models where we're trying to capture as many of the processes as possible and simulate it. And I do a lot of work with climate modelers. And the third thing we do is we try to look for past analogs. These are woefully imperfect because the Earth was not exactly the same. Obviously, what we're doing here when we look at the past is we're trying to look for times when the Earth conducted the same experiment. But as scientists, you understand that the whole point of doing an experiment is that you control all of the variables but one, and we clearly cannot do that in the past. Different vegetation, different critters running around. I always like using this picture because it's so woefully, and paleontologists hate this picture because it's like 100 years old. And look at, that's a slow, sluggish, fat T-Rex there. But I love it, probably because it's the first picture that I ever saw when I was a kid that made me think dinosaurs are cool. I need to bring in a new one with a Brontosaurus now that that's like an official dinosaur again. Anyway, so those are the three options we have. And what I do is I tend to work in the space of the latter two. And I'll show you what that all means as I move on. So what I've not done is I've not actually yet told you what I do. Right? So what I really am is I'm an analytical chemist. I'm a geochemist, sure, I'm a biogeochemist, but really I'm an analytical chemist. I use hyphenated techniques to examine organic compounds and their isotopes in anything. They're all the same types of techniques that we would use in forensics or archaeology or food chemistry, LCMS, GCMS, some fancier things with the isotopes like GCRMS, but I'm interested in determining in a natural system, what are the compounds that are present and what is their isotopic composition? And I generally use that to study things like biogeochemical processes. What are the molecular remnants of microbes in a setting and what do they tell me about how that system works? Or I look at how organic matter is preserved, or I use it to reconstruct paleoclimate. And as you can imagine, these feed into a couple of those things. The work on biogeochemistry, how microbes respond to changes in their environment, that can feed into these Earth system models. And the work that I do in the past, well, that helps us understand what happened during ancient times in response to past global warming events. So I'm going to give you just two brief little stories that illustrate that. But first, I'm going to tell you why I look at membrane lipids, because everybody always wonders, why do you look at membrane lipids? We've got like sort of representative people from the whole school of chemistry here. Who are the organic chemists? No, none. Oh, wow, that's poor, right? Well, all right, so we can talk about it. Well, anyway, if you look at organisms from a natural product perspective, we're magnificent. There's and at Bristol we have a polyketide group that is fantastic, and polyketides are fantastic, diverse, amazing compounds. Or even more fundamentally, DNA is an amazing compound with loads of diagnostic information. So why do I look at the membrane lipids of organisms? Well, there's three main reasons and a fourth special one. So this is a typical membrane lipid that's found in bacteria. This is a hopanoid. This is a type of compound you would look at and think, okay, well, that looks somewhat diagnostic, but it's not. It's not found in all bacteria, but it is found in a very wide variety of bacteria. And let's face it, a tracer for bacteria is not terribly useful. What's nice about the lipids, though, is that although there's these general structural classes, there is variability, especially in the hydrocarbon backbone. We have a methyl group here, or a methyl group here. Just subtle variations, but those subtle variations mean that instead of being diagnostic for bacteria, it's diagnostic for methane-utilizing bacteria, or diagnostic for specific cyanobacteria, which are a particular type of autotrophic algae that live in the ocean. So that means that membrane lipids have a lot more structural variability than you might think based solely upon our classic lipid bilayer model. This structural variability is preserved. During diagenesis, when these compounds are preserved and degraded, you'll lose these functional groups. You might even begin to have cleavage of this chain, but the hydrocarbon skeleton is preserved. And if you don't cook it, by that I mean making oil and petroleum and ultimately gas out of it, compounds like this have been found in rocks 2 billion years old. 2 billion years old, right? These are incredibly robust hydrocarbons. So they're preserved. And then finally, we can easily determine their isotopic compositions. If you look at this type of compound, not the precursor I showed you, but this compound, you think, that looks GC amenable. And if it's GC amenable, I can put it through a gas chromatograph, combust it, in continuous flow into carbon dioxide and water, get rid of the water, send the carbon dioxide into an isotope ratio mass spectrometer. So I know not only the structure of this compound and likely what made it, but I can measure its carbon and also with new methodologies, its hydrogen isotopic composition. So I can learn a lot about what type of lifestyle this organism was having. But finally, of course, and really the reason we look at lipids is that their concentrations are very high. If I want to try to get it out of a sediment or a soil or a rock, can't be looking at beautiful exotic polyketides because these are highly bioactive compounds, which means the organisms don't make lots of them. They do make lots of membrane lipid. So I can analyze this. I can find these compounds in anything. I could find these compounds probably if I were to collect enough dust out of the room. So that's one reason why we use them. Another is that by virtue of being a membrane lipid, we are looking at how microorganisms in particular are mediating their relationship with the environment in which they grow. And that means they are actual tracers of environmental processes in and of themselves. I'm not going to talk about these particular tools, but for example, the distributions of compounds like this in the environment are dependent not only on the organisms living there, but the temperature. So I can look at those distributions in an ancient rock and reconstruct the temperature at the time that was. But I'm not going to go in that direction. I'm going to tell two stories instead that are a little bit more about how we use more novel methodology involving the carbon isotopes of these compounds. So the first story is the anaerobic oxidation of methane. All right, so this is a biogeochemical process that we really only discovered about 15 years ago, although we had circumstantial evidence for the previous 10 to 30 years that suggested that this was happening. But we only nailed it about 15 years ago. And it's a very important process because methane is a greenhouse gas, and we need to feed this into our Earth system models. Okay, so I showed you before carbon dioxide. Well, here's the methane curve, and of course we have a big increase, almost a tripling of atmospheric methane concentrations from about 700 to 1,800 parts per billion. A lot less methane than there is CO2, but a big increase. Largely due. You guys know why there's a big increase in methane? Any guess? That's one. What's the other? Any guess? Sloppiness. All right. So cows, sloppy drilling and gas extraction, just not terribly efficient gas pipelines. Rice paddies are a big one as well. But there's also a natural component. So we know why it's increased. That doesn't quite explain everything, however. And what we don't know is how much of that change is due to perturbation of the natural system and how that might change. By this, I'm talking about horror stories you might have heard about. You melt the permafrost and there's a big bubble of methane that comes out. Or you expand wetlands, or all sorts of different types of processes. That is a wild card in the Earth system. How will the natural system change? So largely, we tend to think about permafrost and wetlands because today, most methane is coming out of wetlands and termite mountains. So mostly it's terrestrial. But the odd thing is that a huge amount of methane is made in marine sediments and none of it escapes. None of it, or very little of it, escapes to the atmosphere. Why is that? Has that always been the case? Will it always be the case? Well, 15 years ago, were fairly fundamental questions. So what we have here is this is the type of record that a geochemist would have collected 30 or 40 years ago, long time ago. And what they see is that in this sediment at the bottom of the ocean, you see this diffusion of sulfate coming down. So the sulfate is being consumed as the terminal electron acceptor in a redox reaction as organic matter is being consumed. The oxygen in this particular core, long gone, rapidly consumed. The sulfate is being consumed, and then the methane is coming up. Or if you look at it from the opposite way, the methane is being generated in the sediments, it is diffusing up, and then is being consumed here at this particular transition. Now that may seem like a rather specific and rather idiosyncratic type of observation, but it's really, really important because what you're seeing here is that the methane is not being consumed by oxygen, which is a highly efficient, very robust energy-producing reaction, and there are diverse and wonderful variety of bacteria that mediate this aerobic methane oxidation that were really, really well understood for decades. But instead, this methane is being oxidized by sulfate. with some pretty important implications in terms of alkalinity and also in terms of whether that methane escapes. Now, the problem is the geochemists looked at this profile and they said, that's sharp, that's rapid, that's not abiotic, that must be a biologically mediated reaction. Microbiologists looked at this. They tried for years to try to culture the organisms and failed and concluded that this was not being mediated by biology. And this is where the organic geochemist comes in, because we have tracers. Now, we didn't have a tracer for this particular organism because we didn't know what it was. So we pulled back and used more generic tracers. Tracers for the archaea. So if you look at this compound here, this looks sort of like a lipid bilayer type compound. Instead of being a acyl compound, however, it's an ether lipid, which is quite typical for the archaea. And here's a lipid monolayer membrane spanning type of compound that, again, is an ether lipid. And we have these types of biomarkers for bacteria, all sorts of things that we can look at. So we can use this to profile what was in the sediments. But we wanted to link it to carbon isotopes. Now, the thing about carbon isotopes is you all know that you have mostly carbon-12 and then a little bit of carbon-13. This is what we care about. These tiny, tiny little variations in the ratio of carbon-13 to carbon-12. Carbon-13 generally makes up 1% of the carbon. Generally. And the tiny variations are all due to biological reactions. And you can use that to trace things. So this is the same thing plotted on what we call a per mil scale. And we're not going to get into definitions of these. But just as you start up here, this is limestone here. And this has generally about 1% carbon-13. As you move down this, you have less and less carbon-13 and more carbon-12. So algal biomass, plant biomass, the tables, organic matter tends to fall around here. In other words, we're lighter than the inorganic carbon on which we live. The methane, way down the scale. So we can use this as a tracer. Organisms consuming methane will look like this. Organisms consuming CO2 will look like this. So we went off to a hot spot. We went off to a place in the Mediterranean where the plates are coming together. It's squeezing the crust. Mud and salt water and methane were all coming out to the seafloor. We went to a hot spot where all of this was happening. Now, I've got a movie here, but I don't think it's going to play because I copied it over. Well, I'll just skip through that. But anyway, it's great because all the movie shows, it just shows lots of gooey microorganisms, which is, I mean, that's what I'm all about, is gooey microorganisms. I mean, yeah. When you spend lots of time in a submersible on the bottom of the ocean, there is nothing on the bottom of the ocean in most places. And then you come across this, and then you bring it up on the deck. And I don't know if you noticed, but this reaction, sulfate being reduced, to oxidized methane, the product is hydrogen sulfide. So you bring it up on deck, and it's not, well, it smells of science, but it also smells of seasickness. Oh, hey! Mute this. Excellent. I don't know who's that talking. So this particular mud volcano we're looking at, this is a place where about 7 million years ago, a big salt deposit was formed in the Mediterranean Sea when it evaporated. So this is an area where the fluids are bringing up brines, they were bringing up methane, they were bringing up all sorts of different materials. And what you were seeing there is black microbial mats sitting on those spots where it's coming up. This is where all of those little brine seeps all flow together into a big river at the bottom of the Mediterranean. And this is a bit of gooey mat that we went and collected here. So it sort of shows you that although it looks like you're looking at just mud, the mud is really quite fluid because of what's been extruded from the bottom of the seafloor. So you see solution. And then that's your nice, gooey, fantastic microbial mat. What you're seeing there is you're not seeing the microorganisms that I'm primarily interested in. You're looking at microorganisms there. You can tell by the white color that they're containing sulfur inside. What these organisms are, they're the organisms that we're consuming. Yeah, I love that picture. Yeah, that's always good for the undergrads as well. These are organisms that are consuming the sulfide. We had to go deeper in the sediment to find the organisms using the methane. I'll come back to why I'm showing you this video in a moment, but what you might think here is I'm not looking at the norm. What we did is we wanted to go and find the hot spot. So again, this was very interdisciplinary. I was there with a lot of oceanographers. I was there with people who studied earthquakes and plate tectonics because they knew where they were going to be found. I worked with geochemists. I worked with microbiologists. We got it all together, and we basically pooled the sample, and we generated lots and lots of data, lots of samples, but the take-home message can be really summarized by a single gas chromatogram. So this is, range of peaks here. And we found all the compounds that you normally find in a marine sediment. In this case, we found cholesterol. This is normally one of the major peaks because cholesterol is in everything. Instead, the dominant peaks were these compounds that come from archaea. Now, that confused us because at the point, archaea were known to be methanogens, organisms that make methane, not those that would consume it. Why they were present in the seeps was confusing to us. We also found all sorts of fatty acids and unusual compounds that were coming from bacteria. So this made us think, like, okay, well, there's microbes living in here. These microbes are clearly consuming the methane. But we couldn't prove it. But by connecting this gas chromatograph to an isotope ratio mass spectrometer, every peak that came out, we were able to determine its carbon isotopic composition. The cholesterol looked like it came from a typical type of algae. Very normal value. These values are lower, they're lighter, they're indicative. They proved these organisms were consuming methane. And what it also proved is it proved that both the bacteria and the archaea were responsible for the methane consumption. And that made the suggestion that what we were looking here was not a single organism mediating this reaction, but rather a consortium of organisms, one of which was archaea operating in reverse, oxidizing the methane with water, forming the bicarbonate, generating a lot of hydrogen. And you can see how thermodynamically this reaction will become rubbish if you accumulate very much hydrogen. But of course, that hydrogen was being consumed by sulfate reducers, reducing the sulfate and generating this hydrogen sulfide that was making me seasick on the boat. So this was quite interesting because if it's a consortium, this explains why microbiologists 15, 20 years ago could not culture it because they could not get a pure culture of the organism that needs to live in consortium. And this opened up a whole new range of collaborations with microbiologists. They did the PCR, they made clone libraries. From this, they were able to do fluorescence in situ hybridization and sort of, color label them. And they could show that the bacteria and the archaea were living in close physical proximity, forming sort of these uber clusters of organisms in the sediment. And they even were able to validate, looking at these clusters, the same isotopic relationships that we did. So we went and we did all sorts of things. There were all sorts of knock-on experiments. One of the things that we noticed is that because this reaction, instead of generating carbon dioxide, which is corrosive, it generated bicarbonate, which is alkaline. What you see is that rather than dissolving calcium carbonate or limestone on the seafloor, you precipitated it. And as a consequence, not only did the methane not escape, but even the CO2 that formed from it did not escape. It was forming these big, thick carbonate crusts. So in a world of lots and lots of bad news about climate feedbacks, overall this was a rather positive story. We learned that the process is mediated by a consortium of organisms. So we learned about how this new biological process worked. Subsequent work showed that this is widespread. Once we knew what to look for, we found it all over the planet in sediments where methane oxidation is happening. We learned that it leads to precipitation of calcium carbonate on the seafloor. And most importantly, this is acting as an additional barrier. Methane is not escaping the atmosphere for a lot of reasons. One is that it's trapped up in hydrates. Then even if it escapes from dissolving hydrates, it not only must pass through this sulfate barrier, but even then it would still have to pass through the oxic ocean without being oxidized. So all of this meant that marine generated methane is really going to be hard to get in the atmosphere. Doesn't mean it's not possible, and there's examples from the past that suggest this, but it gives us some confidence that there's some built-in buffers that moderate this flux and moderate our concerns about this greenhouse gas. All right, so that's one story. I'm just going to wrap up with another story, which is about past environmental change, showing how we can use these compounds when we find them in rocks to say something about some of the things I was saying at the beginning, which is about things like climate sensitivity and such. So these are, this is why I'm a geologist, guys, right? I mean, look, this is where I can do, I don't get to, I'm an analytical chemist, right? These days I'm just in the lab, and the rocks come in a cardboard box. But there was a day When I was a PhD student as a postdoc, I got to go in submarines and I got to go collect samples from places like this. But really what I want to show you here is that you guys see these types of outcrops all the time. Okay, maybe they're not as magnificent as they are here in Utah, but we see this all the time, and we just look and we see layers. These layers are here for a reason. They're forming for a reason. They're telling us the story of how the climate of the past was operating, how it was responding to changes in solar forcing, changes in carbon dioxide, and all sorts of things. And if we can interrogate that record, we can learn something about how the Earth system operates. And basically, this is just an inversion of the principle that Hutton used to found the whole discipline of geology. Basically, his argument was that if you want to understand the past, these rock records, we need to understand the present. We need to understand how things work today. So the present is the key to the past. This is the foundation of geology. But we can flip it on its head and say, well, in that case, we can look at the past and learn something about the future. So what are we doing here? Well, one of the things we learn about the past is that it changes. Climate is variable. We have ice ages. We have times when the Earth was much warmer and there were palm trees in the Arctic Circle and Antarctica. We had times when the entire world was covered by ice. We must not be complacent. Climate does change. Okay, that's all good. But why and how? Can we actually constrain some of those numbers that I opened the talk with? Well, one of the things that we do is we reconstruct carbon dioxide levels, and we reconstruct temperatures, and we try to interrogate this climate sensitivity term. And it's tricky, but we're having some good success so far over the past couple of years. So again, this is the record I showed you before. So this shows you the record of atmospheric carbon dioxide from the ice cores. And of course, it only goes back 1 million years. That's the limit. And that comes with a really critical caveat then to any sort of interpretation of climate sensitivity from the past. And that is, we can only interpret climate sensitivity compared to today when the climate was colder. Nothing in here lets us imagine what climate sensitivity might be like today or far into the future. So we can't test on a warmer planet. We come back to the isotopes. So what the isotopes do is we're going to come back and look at something that I talked about as simply a throwaway line a few slides ago, which is that there's this discrimination between the inorganic carbon on which plants live and their biomass. And it is passed on to all of us. All living organisms have isotopic compositions that look more like this. So what controls this? Well, it's controlled largely by Rubisco, of course. It is basically, you're looking at the isotope discrimination, the fact that carbon-12 reacts faster than carbon-13 in CO2 when it reacts with Rubisco at the first and most important step of photosynthesis and carbon assimilation. But in reality, this is what the world looks like. There's this huge range. So this is the theoretical maximum discrimination. This is what happens when I study Rubisco in a lab and see what types of discrimination it has against a substrate. That is the messy, messy, messy bit of reality. What drives that? Well, we spent a lot of time studying this, and this again goes back, say, 15 years or so. It's trying to set up some of the models in the theoretical framework. Basically, the main thinking at the time was that CO2 was diffusing into the cell. Now, if all the CO2, all of it, is then converted into biomass, there's no fractionation, there's no discrimination whatsoever. Everything that's come in has been used. However, if most of it leaves again, then this Rubisco fractionation can be fully expressed. You get the maximum fractionation. So basically, how much fractionation you have is a function of supply versus demand. How much CO2 is coming in, how quickly it leaves, how fast is the organism growing. So it's going to be all sorts of parameters that affect that. But the main one we want to get out of it is that rates of diffusion will be dictated by the concentration of carbon dioxide. Everything else is a complication. The permeability of the membrane, the diffusion, the fractionation that happens across it, even things like the surface area to volume ratio, which dictates, again, supply versus demand, and of course, the growth rate of the organisms, all of those are going to dictate what we observe, and we want to constrain those and pull out CO2. The problem is that algae are wildly different. They have different shapes, they have different geometries, they have different sizes, they have different membrane permeabilities, they have different growth rates, even when growing in the same environment, all sorts of variation. So what we want to do is not measure biomass of all organisms and CO2. We want to measure the biomass of a really narrow, specific group of organisms from which we can constrain all of those extraneous factors. And we use this organism here. This is a Miliani huxleyi, which is a beautiful, beautiful, magnificent organism. I put up here. This actually is the logo of the Phycological Society. That's how much they love this. It's fantastic. Mammoth blooms happen around Ireland and Britain of these organisms. They form vast white clouds. They're amongst the most important organisms on the planet. And fortunately, for reasons unbeknownst to us, they make a rather diagnostic compound. This is not an interesting compound like some of the others I showed you. It's a C37 or C38 long-chain alkyl compound that happens to have 2, 3, or 4 double bonds and a ketone at the secondary carbon. In other words, it's a long-chain unsaturated ketone. But that combination is unique. It's unique in the biological world, which means if I measure the isotopic composition of this, I'm measuring the isotopic composition of this organism. And if I measure the isotopic composition of something else that is tracking the CO2, I can now determine that fractionation. And I can use that to reconstruct the carbon dioxide concentration in the ocean. And then applying Henry's law, I can use that to reconstruct the partial pressure of CO2 in the atmosphere. I'm going to skip over this. One of the things that I've been particularly keen on is not leaning on only a single proxy. One of the main characteristics of my own work over the past decade has been working with a lot of people who are a lot smarter than I, using other proxies and pulling them all together and telling a somewhat consistent story. And this particular proxy is based upon looking at boron that has been partitioned in the calcium carbonate of shells and looking at its isotopic composition. And its isotopic composition is pH dependent. And if you know pH, you can then make some assumptions about alkalinity and again, back out CO2. So all of this is culminating in this really, really messy slide, because when you use lots of different proxies and you study the past, it gets messy. So I'm going to walk you through it very slowly, starting with this plot here on the bottom. And this is showing effectively the descent into our modern ice age climate. What you see here is, down on the bottom of this axis, this is oxygen isotopes in foraminifera. You get these low values when it's cold, you have a lot of ice, and you have a low sea level. In other words, this, those are ice ages. And what you see here in the black, those are the records from the ice cores that I was showing you before. So you're seeing the CO2 variation from about 180 to about 280 parts per million during these ice ages. So we wanted to reconstruct carbon dioxide concentrations going back further in time. And what you get is a mess. Why do you get a mess? Well, you get a mess because biology is wonderfully and magnificently messy, right? Biology is not working simply. We have to make assumptions about the growth rate. We have to make assumptions about the algae making it. We're looking at not just a single core from the Atlantic Ocean and making all our interpretation based upon that. We're looking at cores from all over the planet. And we're using completely different techniques. We're looking at leaves and the number of stomata on them. We're looking at these proxies that I was just telling you about. And we're pulling them all together. And of course, when you do that, science gets messy because we're not going to tell a story based upon a single one of those lines. That's all very noble, but can we actually then say anything from it? Well, actually, we know that this part of this time in Earth history was markedly warmer than today, about, say, 1.5 to 3 degrees higher global temperatures. So it's warmer. And yet, we can say with some degree of confidence that CO2 was probably not much above 450 parts per million, and you can even have some degree of confidence that it was not even above 400 parts per million. Carbon dioxide was higher when it was warmer, but it wasn't a lot higher. And we can use that then to, again, interrogate climate sensitivity. And that's what this figure shows here. This little cross is at 0 and 0. What you see on the x-axis, this is the forcing, largely from CO2, but there's another factor in here which I'll ignore right now. This is the forcing in watts per meter squared, and this is the global change in temperature. So obviously today, before we started burning fossil fuel, we're at 0. There's 0 change on either axis. This bit is taking us into the ice ages. You have less forcing, you have a colder climate. And the blue and the green is taking us into the past before the ice ages. And again, if you have more forcing, you have a warmer climate. Looking all the way through this, you might be able to imagine some inflections, some changes and all sorts there. But the real take-home message is that when it was colder and it was warmer, the climate sensitivity was about 3 degrees. Exactly what climate models predict. Entirely consistent with all of our forecasting for the future. Now, of course, this still comes with a probability distribution function because there's a lot of error and range in both of those. And there's error in some of the plots I was showing you before. So we've not actually narrowed the range for climate sensitivity, but we've increased our confidence in it by looking at these past examples. So obviously there's a lot more that we want to do. In particular, I don't want to just look at CO2 and warming. I want to look at glaciers. I want to look at the acidification of the ocean. I want to look at how life has responded. We do all of these things. How did our planet respond when it was warmer? How was it different? Were crops, was vegetation, did it become nitrogen limited on a high CO2 world? Were organisms living in the ocean stressed because it was more acidic? How much does timing have to play in this? So there's a whole raft of experiments that we can do by looking at climates that are much warmer than they are today. Just sort of summarize what we sort of have pulled out in all of this. Probably, I think, the most important thing I do like to tell people is that Earth's climate does change, but it changes consistently with our overall understanding of the climate system. Climate and the chemistry of our environment right now are changing faster than they've ever changed in the past, however. That does not come across in what I've talked about today, but you will rarely see what we're doing today happen in the geological past. The knock-on effects are complex, but I think that chemists are going to play a particularly crucial role in understanding this complexity. We need to interrogate these processes at a fundamental and mechanistic level. We can't just look at what happened over the past 100 years and propagate those trends into the future. We need to mechanistically understand the chemistry of soils, the chemistry of the oceans, and the life that depends on them. And also, I feel like putting this in here, sort of reflecting upon previous talk and reflecting on what you want to do, is that actually all I do is I talk about the problems. I try to help with understanding the problems by getting a better understanding of what will happen in the future. But if you want to think about food security, if you want to think about new materials, you want to think about new electrical innovations in energy, effectively that is where chemists are needing to lead the way. And I do think that some of the most exciting things about being director of the Cabot Institute has not just been going off and working with social scientists and engineers, but actually having new interactions and new relationships with the chemists in my own department. Because chemists are really right at the forefront of coming up with the exciting innovations that will address some of these global challenges. With that, I will put up various, just one of these. Yeah, I've been quite lucky. I've worked with a lot of people, and they're all apps. Look, when you're all older, you'll all have slides like this, right? But most importantly, I wanted to thank you and the RSC for inviting me here and letting